First of all, I want to thank Dr. Shazia for inviting me here. I know very few people here because maybe they are not interested in basic science. But I want to tell one thing, that without making a good radiopharmaceutical, you cannot do good imaging. Whatever camera you use, if you don't make a good drug, you will not get good imaging. So I think basic science has a very critical role in nuclear medicine. So I think there should be interest from physicians also to take interest and participate in this session. So KFSH, King Faisal Hospital, is a tertiary care hospital in Riyadh. We have 1,600 beds. We have more than 10 cameras, very modern. And on top, we have 3 cyclotrons that we built about 40 years ago. And we produce and we have about 15 hot cells where we do all GMP practicing. And we produce about 30 radiopharmaceuticals. They are both diagnostic and therapeutic, including technetium generator. And we supply these to the 50 hospitals in the kingdom. On top of this, we produce FDG 3 times a day, about 5 curie. And we supply 50 hospitals and 150 units every day. So our department is a service provider, and also we have a small R&D department where we make new drugs. So we make, for example, fluoroestradiol, PSMA, ammonia, FDG, choline, DOPA. So all different types of PET isotope that you can imagine. On top of this, we can make fluoride, FLT, dotatate, PSMA with lutetium and with gallium. As we are talking a lot about peptides, so what are the peptides I will discuss briefly in this presentation. You know this dotatate they are talking. Basically, dotatate or somatostatin analog is 28 amino acids. I don't know if So this 28 amino acid is not stable in the body. So that's why they make analog, short analog of 8 amino acids, that you can see that this 8 amino acid has dicysteine bond. And initially it was labeled with indium. So the idea is to make analog that can be used as a theranostic, can be labeled with DOTA and also with lutetium and gallium. That's why they replaced DTPA with DOTA. And another isotopes recently approved by FDA is copper labeled dotatate, which is DetectNet. The idea is to make it longer half-life because copper is PET isotope, but it has 12 hours half-life. So the shelf life of this isotope is 24 hours. So it can be shipped outside the country, another cities, and it has more better that you can do late imaging also. As I mentioned that we have mainly service department, but on top we do preclinical evaluation. I don't know how it works. So these preclinical means we can do peptide synthesis, we can do animal imaging, we can do labeling, all types of things that need to be drugged for a new drug development. Why peptides? So I want to emphasize, compared to antibodies, peptide has different properties. For example, they are small in size, they can be synthesized chemically, and also they are easy to radiolabel because they have a small chain. So any isotope or any chelating agent can be attached. Because of the small size, they have rapid kinetics, they are rapid target issues, and also they are non-toxic. On top of this, they have a lot of receptors that can be used for targeting. So these all properties make peptide ideal radiopharmaceuticals. On our lab, we have interest on different types of peptides. For example, bombacin, because bombacin is a small peptide. Then we have mucin peptide. We have HER2. HER2 is derived basically from Herceptin, which is an antibody. Then we develop also leuprolide peptide, which is for breast cancer imaging. We also conjugate PSMA with different peptides to enhance targeting efficiency. On top of this, we also conjugate peptides with cytotoxic drugs, for example, doxorubicin, also with folic acid, also we work on rhodamine. So our activities in the lab make us to be more useful making new drugs for future applications. The one thing I want to emphasize that we also work with cardiac imaging, and in this context, we make rhodamine. As you know, rhodamine is like a amoeba, And we try to make it with nota and label with copper because of the longer half-life. And as you can see, a very nice heart imaging here. And based on this discovery, I got this American fellowship because they think it's a useful compound for future application. So when we design a peptide, what is the criteria? The criteria is first you have to know the target, which target you want to target. For example, for breast cancer, it's a different sequence. For prostate cancer, it's different. Based on your target, you design your peptides, and then you attach a linker. Linker can be amino acid, can be any molecule, and also depends on your application, whether it's diagnostic or therapeutic, you have to attach with that one. So all these criteria, you have to plan ahead before you design a peptide. For example, somatostatin receptors, you know that you are targeting neuroendocrine tumor. So then you have to attach your somatostatin or octreotide analog here. Then you do your linker and attach with, for example, DOTA. DOTA has the ability that it can bind with any isotopes. You can use lutetium, you can use copper, you can use zirconium, depending on your availability of the isotopes. So all these making isotopes for theranostic, diagnostic, all depends on your molecule that what you want to apply. So our interest in our lab that we do our own synthesis. So we have this facility that we can synthesize our own peptides. And for this peptide synthesis, we don't need a machine. We can just simply use a funnel, which is very simple, that you simply use resin, then you attach your amino acids, and at the end you attach DOTA. So you can make your own peptide depending if you have this availability in your facility, and need also a little bit experience and technical information. So our interest also, because peptide alone sometimes is not enough, so you have to add some doxorubicin, some anti-cancer drugs. So peptide can be used as a carrier. So when you use carrier, then you can use any drug depending on your application. So we use this receptor binding peptide as a carrier to take these cancer drugs to the target. In this way, it's more specific, less toxic than conventional chemotherapy. Also, we try to use different peptide cocktail. As you know, only one peptide sometimes is not enough. So you have to use peptide cocktail, or you have to use 2 different drugs conjugate with peptide like antibodies, and you can use nanoparticles. We are trying also estradiol because estradiol is ER positive. So the idea is to make dual targeting, so you have more receptors, and so your targeting efficiency will be higher. As you know, gallium is available in the form of jet generator, as in technetium, but because gallium has 68-minute half-life, so our idea is to produce gallium from cyclotron. In this way, you can make more drugs, more doses, because with generator, you can get only 50 millicurie, but with cyclotron, you can make up to 500 millicurie. And if you have high demand of gallium dotatate or gallium PSMA, in this way, cyclotron production is very important. That can allow you to make multiple doses. In terms of gallium kit, it's like a technetium kit. So you have your precursor available in that vial. So you have to adjust pH 3 to 4, you heat it, then you do SEPAC purification to remove unreacted gallium, and then you have pure sterile product of gallium-68 product that you can use for the patients. Why I'm telling you, because I noticed that some people complain about, for example, imaging, making extra doses. So I think first thing you have to make sure that whatever you are injecting to the patient is up to the standard. So you have to do all quality control specification test before you can make sure that what you are injecting is up to the standard, and you can expect good imaging after injecting good radiopharmaceutical. So for the newcomers, labeling is very simple. So you just have your peptide, you just adjust pH with sodium acetate, you add gallium and simply heat. So these are very simple reaction for gallium labeling. Same way you can do for lutetium. In lutetium, you use also gentisic acid, which is anti-ascorbic agent. Because lutetium is high energy, it forms radiolysis, so you have to prevent that radiolysis. That's why you have to add some gentisic acid. What is theranostic? People are talking theranostic, but the idea of theranostic is to use one drug for both diagnosis and therapy. For example, you use somatostatin, so the drug is same. The only thing you are changing is the radioisotopes. So in this way, you don't have to get two approvals from FDA. One drug can be used for the approval. So the same peptide can be labeled with, for example, if you want lutetium for therapeutic, gallium for diagnostic, copper 64, zirconium 89. So all these possibilities are available for labeling with the same peptide, and they use two purpose. We are working also with PSMA. As you know, PSMA is highly expressed in prostate cancer, and its expression is about 1,000 times higher. So whenever you make any drug for the receptors, you have to make sure that it is highly expressed on receptors and low expression in normal tissues, because you don't want radiation to the normal tissues. PSMA in this respect is useful because it's highly expressed. As you know, for prostate cancer, prostatin is the antibody, but we are not interested in antibody because it's expensive, larger size. So out of antibodies, we make a small molecule called glutamate. So as you can see, this molecule, this is a very small molecule that can be used for PSMA, and these contain urea base. And you have this amino group that can be attached to any molecule like peptide to make it linker. In this way, you can synthesize your own PSMA. In our lab, because we want to use PSMA for different isotopes, so we make technetium version of PSMA, we make copper version of PSMA. So in this way, we do our own chemistry. So we start, for example, I don't want to go in detail. So we can use, for example, DOTA for copper, 64, gallium. But on top of this, we also make it fluorine version, because fluorine has longer half-life than gallium. So you can make more doses. Also, on top of this, you can make it technetium version. So in case people has no gallium, no fluorine, they can use also technetium version. So what you do, this is your basic unit that binds with the PSMA receptors. Then you can use linker, and then you can attach this chelating agent, which also can be synthesized on peptide synthesizer. These are simple amino acids that can be used. So PSMA, as you saw in yesterday's lecture, can be used more than prostate cancer. So we are trying to do preclinical testing of PSMA in triple-negative breast cancer. We do some animal studies, and this work is in progress. I don't want to discuss because these are under patent. So our interest mainly on bombesin peptides. Why bombesin? Because bombesin is 7 amino acid peptide. It is very linear. And you can see this 7 amino acid peptide, that this side is free. So you can attach any linker, any chelating agent here. So you can use for lutetium, gallium, technetium, fluorine, depending on your application. So the idea of peptides that it are flexible. So you can do a lot of modification based on your requirement. And also, bombesin receptors are expressed on prostate, lung cancer, breast cancer. So it's very unique peptide that can have further application. Another peptide we are working is mucin. As you know, epithelial cancer express a lot of receptors for breast cancer, ovarian cancer, and this has low expression. So we are also trying to label this peptide with different isotopes for further application. Also, we are working on HER2 peptide derived from Herceptin. Herceptin is an antibody FDA approved for breast cancer, but we are more interested as a shorter version. So we take this peptide for further application in breast cancer. Also, folic acid, because folic acid is expressed on breast cancer, and methotrexate is anti-cancer drug. So these compounds can be labeled with drug, peptide to enhance targeting efficiency. Fluoroacidiol is another approved compound that can be used for ER-positive cancer. So we are making fluoroacidiol as a DOTA because we want to label with lutetium also. So we are trying to make some changes. Another isotope which is very useful for future application is zirconium 89. As you know, zirconium 89 has long half-life, is a PET isotope, can be used for antibodies, can be labeled, and then you can do imaging next day. This is the first PET isotope that can be labeled and used after 2-3 days. So it has very potential application. So we are working on the production of cyclotron produced zirconium-89. In this way, you need this energy. Then we have to confirm the identity. When you make a new compound, you have to make sure that this compound is pure. It can be used for the patients, and it contains no impurity. And then we do all, okay. So we do all this purification to make sure. Then we do radio label. We label with DOTA PSMA, and you can see it's very clean product. And also then we do animal studies to make sure that we are using for targeting. And also we do PHCT, animal. And then we have to make sure all these tests before we can inject to the patient, that the safety is very important, quality is very important. You have to meet all these specifications, make sure that this peptide is good enough. So we do all these clinical testing. I'm not going in detail. And then you can see that our steps are very, very extensive that we do. I want to emphasize one thing, that people who do research, they start maybe from 5 to 10,000 compounds. And it takes 10 years to make one compound. From 10,000, you can make one compound. So never give up. You always do keep good research. And I want to give you one result. So recently, this is for encouraging only, not for publicity, that I was top 2% because of if you keep doing good research, one day you will be rewarded. So always be hopeful, because research always very frustrating. You always get upset, but if you keep working, one day you will be rewarded. So our interest mainly is to translational research from bench to bedside. And finally, I want to thank all for listening. Thank you.